Back in 2011, there was a media storm when researchers in Brazil identified four different types of brain-seizing fungi that each infest a different species of carpenter ant. The ants become hosts when they ingest Ophiocordyceps spores, which proceed to germinate in their thorax. The fungus spreads through the ant's body, sending spiny tubules into its abdomen and head. The fungus hijacks the ant's neural and hormonal pathways, enslaving the host to the parasite. Their bodies break down over the next 4 to 10 days, and the infested ants shamble chaotically around the nest, dropping spores and sometimes mistaking their own nest mates for food. In their final delirious hours, the ants climb down from their trees onto the jungle floor, where the cooler, more humid climate is more hospitable to fungus. They climb onto short pieces of vegetation and latch on underneath. And over the next few days, the fungus's reproductive hyphae bursts through the base of the ant's skull. The fungus lives on, consuming the ant's remains as it disperses its spores toward the soil. And as crazy as all this sounds, there's even a second fungus that preys upon the first one. The carpenter ants in Brazil aren't the only ones that can become zombified. And these researchers at the University of Copenhagen, Simone Gasca and Brian Fredensborg, just published a study on another species. And these ants live in the Bidstrup Forests of Denmark. And while the parasite and the ultimate fate of the hosts is different, it's every bit as fascinating. I want to talk about the ants and the parasite that infests them. We'll talk about its life cycle and how it parasitizes the ants and other organisms along the way. I'll discuss the researchers' findings about the zombified ant's behavior and the environmental cues that trigger it. We'll also look at how these things lead the ants to their ultimate fate. And finally, I want to touch on some things that are still unknown. So let's get into it. I'm Cameron and this is my channel. Welcome to the Sexy Universe. There are 3 species of European redwood ant, and between them they cover most of Europe and Asia. The ants the researchers looked at were members of Formica Polyctena, a species that's almost entirely found in cooler northern climates. They live in colonies of genetic clones, almost all of which are sterile female workers. Males and queens of this species have exclusively reproductive roles, and because queens can mate with genetically identical males, and produce eggs that also become queens, colonies can have multiple queens and spread across multiple nests. They recognize their nestmates and distinguish between them and outsiders through their unique pheromones. These ladies build enormous nests compared to what we see in the United States. They can easily measure over 5 feet tall, and some get as high as 20 feet. The ants get most of their nutrition from farming aphids. The aphids consume sap from vegetation, but in order to get enough minerals, they have to consume an enormous amount. They secrete the excess sugars as honeydew, and this sweet liquid is coveted by the ants. The ant colonies also have dedicated groups of workers that act as foragers, and they scavenge other food sources from the forest. One food they particularly enjoy are slime balls, a mucousy waste that snails eliminate through their pneumostomes. The pneumostomes are their respiratory pores, like the one you see here. And while the slime balls might not seem like a delicious food to you and me, the ants seem to love them. Lancet liver flukes, Decrocoelium Dentriticus, were described by Carl Rudolphi in 1819 in his text Intozoarum Synopsis. There were earlier mentions of the fluke, but this was the first medical text to describe them. He described their adult phase, where flukes lodge in the gallbladders and bile ducts of cattle and sheep, along with those of wild ruminants like deer and elk. They've also been known to infest humans and other mammals, but this is relatively rare. It's typical to have several flukes, possibly even dozens of them, living in the same animal. They feed off the host's liver cells and shed eggs into its intestine. And the flukes can reproduce sexually with other flukes or asexually by themselves. So shout out to Miley Cyrus, because she can buy herself flowers, but these flukes can do more than that. Lancet flukes can be found throughout Europe, Asia, and North Africa, but are only sporadically found in North America. The eggs pass through the intestine and enter the environment through the animal's waste, and snails like to feed on that waste. Lancet fluke eggs mature and hatch in the snail's intestine, and the next step of the life cycle is called Miricidia. The Miricidia penetrate the intestinal wall and lodge in the snail's hepatopancreas, which is the snail's equivalent of a liver. There they develop into Sporocysts, which replicate themselves at least a couple of times before moving to the next step of the life cycle. The Sporocysts feed on the snail's tissues while cranking out what are called Cercariae. The Cercariae are the next step in the fluke's life cycle, and hundreds of them can be produced. The Cercariae migrate to the snail's lung, and the snail's body responds by coating them in slime and expelling them through the pneumostome. The whole process within the snail's body takes 3 to 4 months, and it can only happen when the environmental temperature is at least 40 degrees Fahrenheit. So these snails are expelling slime balls with Cercariae inside them. And even though it's obvious to us that the ants shouldn't eat them, it isn't so obvious to them. And as you've probably noticed, each stage of the fluke's life cycle depends on it being eaten by the next host. And it progressively finds ways to attach itself to something the next host finds delicious. As Mary Poppins said, a spoonful of sugar. And they also produce several clones of themselves at every step. When the Cercariae enter the ant's intestine, they latch on as the fluke does at every stage, and they lose their tails in the process. At this point, they start probing the surrounding tissue, hunting for the esophageal ganglion, a major nerve center in the ant's head. The first one to reach it latches on and becomes what's known as a brainworm. One interesting thing about the brainworm is that it never develops the protective coating it would need to survive in the next host. So in a strange way, it's both cruel and altruistic. From this spot, it's effectively able to hijack some of the ant's behavior, zombifying it at will. The rest of the Cercariae transmute into Metacercariae and migrate to the ant's abdomen. And once they're there, they do the same thing that happens at every stage of the fluke's life cycle. Feed on the host's tissue from the inside while producing clones of themselves. The encapsulated ant in this photo died with her abdomen full of Metacercariae, and as you can see, they've burst through her exoskeleton. But killing the ant from the inside isn't the fluke's goal. Its goal is to get into the bile duct of a mammal, and the zombified ants are forced to help. It was first documented in the late 1960s that ants infested with Lancet liver flukes have a habit of latching onto blades of grass in the morning and evening. It was assumed but never verified that they were driven by changes in an internal clock or by sunlight. But the researchers who wrote this paper wanted to find out exactly which environmental factors drive the ants' tetany. They observed 1,264 zombified ants on 13 different days that were spread out over a year. The environmental factors they measured were temperature, relative humidity, time of day of the year, and the amount of sunlight. They didn't rely on weather reports. Instead, they measured temperature, relative humidity, and solar irradiation at the point where the ants were observed. Of the original set of 1,264 ants, they marked 172 of them by gluing numbers to their abdomens. This allowed them to keep track of individuals throughout the day. To verify that the zombified ants were actually infested, they captured 176 of them, took them back to the lab, and dissected them under microscope. 97% of them were found to have Metacercariae in their abdomens. A control group of 80 ants were collected from the top of the nest. None of them were found to be infested. Of the factors the researchers measured, only temperature and relative humidity correlated significantly with latching onto grass. The relationship between temperature and compulsive biting was negative, so as temperatures rose, fewer ants were found latching to vegetation. And the correlation was about 11% less biting behavior for every increase of 1 degree Celsius, or about 6% less for every degree Fahrenheit. It was the opposite with relative humidity, though the association with relative humidity was much weaker. When relative humidity was higher, more ants were found stuck to vegetation. However, relative humidity and temperature tend to change in opposite directions. And this is because the capacity of air to hold water vapor increases when temperature increases. Since the vapor capacity changes faster than the actual amount of vapor in the air, relative humidity tends to change inversely to temperature. So the researchers concluded that the significance of relative humidity was actually a mirror of the temperature effect. And this is also supported by some of their other data. The experiment had 5 observation days between September 15th and October 12th of 2016. And since that stretch was in the early fall in Denmark, the daily temperature dropped from 77 degrees Fahrenheit all the way down to 45. So on this graph, we can see that the dotted line at the top represents relative humidity. The dashes near the bottom are temperature. And the solid line represents the percentage of zombified ants that were seen biting onto vegetation throughout the entire day. So these ants bit on and they didn't let go. And you'll notice there's an obvious correlation with temperature, but relative humidity doesn't really seem to have any effect. And here's a second interesting thing. The larger the daytime temperature swing, the more cycles of latching on and letting go the ants went throughout the day. So they'd bite on, let go, and then come back later to do it all over again. And the smaller the temperature swing, the more likely they were to just bite on and stay there. So as they show here, on the day with the 45-degree temperature range, the 8 ants they observed went through an average of 4 cycles of biting on and letting go throughout the day. But on this day with the very small temperature range, the 12 ants they observed, on average, only went through 3/4 of a cycle. Large temperature variations in a day tend to correlate with hotter temperatures. So on days when it was hotter, the ants didn't just stay up on the vegetation. They went up, latched on for a little bit, then let go and went back down to the cooler forest floor. And the researchers believed it's just because high temperatures are not as survivable for the ants or the parasites. On cool days, however, many of the ants never came down at all. And based on this, the researchers believe that the ants climb up onto vegetation and latch on in the early evening and then stay attached throughout the night. When the sun rises and the temperature warms up, they climb back down and get back to work. And since ruminants are most likely to be active and grazing in the hours around dawn and dusk, this gives the ants the best chance of being consumed by the next host. When the ants are eaten along with the grass, the brain worm is digested while the Metacercariae in the abdomen attach to the intestine. Eventually, they migrate to the animal's bile ducts, where the cycle starts all over again. There are several questions that still remain. Attaching behavior isn't entirely foreign to ants, because they can do it when they sleep. But attaching onto vegetation in the outdoors and staying attached is strange behavior for them. And the chemical pathway hijacked by the parasite hasn't yet been investigated. The Ophiocordyceps fungus uses the Parasite-Encoded protein tyrosine phosphatase pathway. This pathway activates the takeout gene and juvenile hormone in carpenter ants, which are related to their internal feeding clock. This leads to a cascade of hormones and neurotransmitters that allow the fungus to hijack the ant's behavior. So it's entirely possible this pathway is also used by the Lancet fluke. So the researchers suggest investigating this pathway first. The researchers also noted that in April of 2017, 5 times as many ants showed zombified behavior as had in October of 2016. So they believe that many ants were infested during the late summer and early fall, and the parasite was able to take over while they wintered in their nests. And while this seems like a probable explanation, we can't be certain of it until it's confirmed by observation. So on cool summer evenings, when you find yourself up on a hill or rooftop, looking up toward the darkening sky and wondering when our friends from outer space will finally come for you, Spare a thought for our tiny cousins who are latching onto blades of grass and waiting on a much darker fate. I believe the ideal life is a non-stop process of learning and growing. Thanks for learning and growing with me. As always, the paper will be linked in the description, as will the show notes, which are available on my website, thesexyuniverse.com. I want to hear what you have to say, so please put your thoughts or information you'd like to share in the comments section below. And hit the like and subscribe buttons on your way out. Stay on your game, and I'll see you next time on The Sexy Universe.